Hi, I am Dr. Arsalan and today we are going to discuss the preparation of culture media or growth media for the growth and proliferation of microorganisms. In this lecture, we will learn the preparation of media theoretically as well as practically in our lab. So media, it is the mixture containing all the essential nutrients required for the growth of bacteria and other microorganisms. So media is basically balanced diet for the microorganisms. It contains all the essential and vital nutrients required for the growth of bacteria. For example, water, amino acids, proteins, carbohydrate, vitamins, enzymes, each and everything is present in the media and is required for the growth of the bacteria. So the bacteria can easily be grown on the media. So this is basically the food for bacteria. In definition, we see there that bacteria and other microbes. The other microbes include fungi, yeast, and molds. There are three states of the media: solid, semi-solid, and liquid. And on the basis of these states, there are two types of media: the broth and agar. Broth is the media in liquid form. It is prepared in test tubes, while agar is the media in solid form and is prepared in the petri dishes. In our today's practical, we will prepare the agar. For preparation of the media, you need these equipments and chemicals in your lab. These are media is commercially available in the powder form. So it is available commercially in powder form and it needs to be reconstituted in the distilled water. So for preparation of media, there are two chemicals, the media and distilled water. Media is available in the packs of 500 grams of bottle and all the instructions of preparation of that media are available on the description on the bottle. The second is distilled water, which is required for the reconstitution of media. Third one is the conical flask. You have to measure this distilled water in the conical flask of 1 liter. And then the beaker. Beaker is required for the reconstitution or mixing of the media in distilled water. Physical balance is required to weigh the media. Stirrer is used for mixing of media in the distilled water. So media is mixed in the distilled water using a glass stirrer. After stirring and mixing the media is boiled on the gas burner. So gas burner is required for the boiling of media in the beaker. Then this boiled media is sterilized in the autoclave. Autoclave is used for sterilization of the media. It has 121 degree centigrade temperature with 15 pounds of pressure and 15 minutes time is required to sterilize the media completely from the bacteria as well as from their spores. The media is poured in the petri dishes. These are the glass type petri dishes and then the media is cooled in these petri dishes. After cooling the media, it becomes solidified and it's called agar. So agar is prepared in the petri dishes and is incubated in the incubator. Incubator gives temperature of 37 degrees centigrade. So the media is then placed in the incubator. In this way, media is prepared. After preparation of the media, the bacterial cultures or bacterial samples are inoculated or loaded on this media and this media is incubated for 24 to 48 hours. After 24 to 48 hours, the growth of bacteria is seen on the media. Physically, their colonies are visible and then you can confirm the bacterial growth by preparing slides and visualizing under the microscope. The last thing is hot air oven. Hot air oven has temperature of 180 degrees centigrade and we sterilize the glassware for 40 minutes. So 40 minutes are required for the glassware. Preparation of media, you need pre-sterilized glassware. So it is required for sterilization of glassware. For example, the conical flask, the beaker, and the stirrer, the petri dishes. These are the four components of the media preparation which need pre-sterilization in the hot air oven. As I've told you that the media is available commercially in the powder form in the packs of 500 grams of bottle. We also show you in this lecture. The most common used media in the lab are nutrient agar and mannitol salt agar, also called MSA, and the McConnell agar. Nutrient agar is used for the growth and proliferation of any type of bacteria, fastidious, aerobic, anaerobic. All these type of bacteria can be grown on the nutrient agar. This is general media. Second one is mannitol salt agar. It's also called MSA and it is selective media for Staphylococcus aureus bacteria. Only Staphylococcus aureus bacteria can grow on this media. And third one is McConnell agar. McConnell agar is used for the growth of Escherichia coli bacteria. Escherichia E. coli. So all these type of media can be prepared as per description of the manufacturer or instructions of the manufacturer which are available on these packs of the media. For example, in case of nutrient agar, you need 28 grams of nutrient agar to be dissolved in 1 liter of distilled water. In case of mannitol salt agar, you need 111 grams of the MSA media to be dissolved in 1 liter of distilled water, and similarly the McConkey agar, 50 grams of the McConkey agar is needed to be dissolved in the 1 liter of distilled water. So this powdered form of the media is dissolved in the distilled water as per instructions of the manufacturer, that how much weight of the media to be reconstituted in the distilled water, as I have mentioned here. Now the last and most important step of media preparation is the procedure of media preparation. As we know that the media is available in powder form, so you have to weigh the media as per manufacturer's instructions, and today we are going to prepare the nutrient agar. For nutrient agar, we have to weigh 28 grams of the media in 1 liter of distilled water. So the media is weighed on the physical balance. The quantity of desired distilled water is measured in the conical flask. Different type of conical flasks are available in the lab. 500 ml or 1 liter of conical flask, these are used to measure the distilled water. Both of these media and distilled water, these are mixed in the beaker. After mixing, the glass stirrer is used to stir the media in the beaker. After media stirring, it is boiled on the gas burner. It's boiled for a few minutes on the gas burner so that the media is completely dissolved in the distilled water. After complete dissolving the media, it is sterilized in the autoclave. Autoclave is the most vital equipment of microbiology lab, in which you sterilize the liquid form of mixtures or media. So this media is sterilized at 121 degrees centigrade temperature, 15 pounds pressure for 15 minutes. Clave decontaminate the media from bacteria as well as spores of the bacteria. After decontamination, the media, it is poured in the petri dishes and allowed to cool. When the media is cooled down, it becomes solidified and it is called agar. The solid media is called agar, and this agar is maintained in the petri dishes in the incubator. This agar is maintained in the petri dishes in the incubator, and these plates are then shifted to the incubator where there is 37 degrees centigrade temperature, and then the agar is ready for bacterial culture. Then the bacterial samples are loaded or inoculated in this media, and this media is incubated at 37 degrees centigrade in the incubator for 24 to 48 hours. After 24 to 48 hours, you can clearly see the growth and colonies of the bacteria. The same bacterial colonies can be picked up and slides can be prepared for the microscopic examination and for confirmation of the bacteria. Now we will practically demonstrate the media preparation in our lab. The most commonly used media in the lab are mannitol salt agar, McConkey agar, nutrient agar. And you can see here that on mannitol salt agar, the composition method is given in the description, that 111 grams of the MSA should be dissolved in 1 liter of the distilled water. Similarly, McConkey agar, it needs 50 grams to be dissolved in 1000 ml of distilled water, and nutrient agar, 28 grams of the nutrient agar is required to be dissolved in 1 liter distilled water. Now I am going to prepare nutrient agar, and as per description that 28 grams of nutrient agar should be dissolved in 1 liter of distilled water, I am going to prepare half liter media. So we can calculate here that 28 grams of media can be dissolved in 1 liter, and for half liter we will need 14 grams of the nutrient agar media. So we will take 14 grams of nutrient agar media and 500 ml of distilled water. This is the physical balance, and we are taking this beaker. This is 101 gram, so tear it, it will become 0. Now put 14 grams of the nutrient agar as we have calculated. So I have put 14 grams of nutrient agar in this beaker. As you can see, this is the nutrient agar. Now I will take 500 ml of the distilled water. This is 500 ml distilled water in the conical flask. Now I will pour this distilled water in the pre-weighed 14 grams of the nutrient agar. Stir it. This media will be stirred using a glass stirrer. After proper stirring, the media was boiled on the gas burner. After boiling, it was autoclaved at 121 degrees centigrade and 15 pounds of pressure. After autoclaving, we will pour the media in the petri dishes. After pouring the media in the petri dishes, these petri dishes will be allowed to cool, and after cooling, these will be incubated. Now the media has been cooled down and it is in solidified form. This solid form of the media is called agar, and it is now ready for bacterial inoculation or culture. Now we place this media in the incubator. And this media is ready for bacterial inoculation.